For example, let's take a look at how the incretin system helps control postprandial blood glucose levels. After we consume food, increased luminal levels of carbohydrates, fats, and proteins rapidly induce the release of various peptide hormones and signaling factors into the bloodstream from enteroendocrine cells further along the gut. Two of these hormones, glucose-dependent insulinotropic polypeptide or GIP and glucagon-like peptide 1 or GLP-1, have been shown to stimulate insulin release from the pancreas. GIP is released from K cells, which are predominantly located in the proximal small intestine, whereas GLP-1 is produced by L cells, which are primarily found in the distal small intestine and colon. GIP and GLP-1 travel through the bloodstream to the pancreas. In the presence of glucose, these hormones bind to a G-protein coupled receptor on beta cells, resulting in enhanced insulin biosynthesis and secretion. In healthy individuals, incretin hormones may be responsible for between 50 and 70% of the insulin released after a meal, although this point is still a matter of some debate. The increased levels of insulin enhance glucose uptake by the liver, skeletal muscle, and fat tissue, and lower the concentration of circulating glucose. GLP-1 also inhibits the release of glucagon from pancreatic alpha cells. Lower plasma glucagon levels signal the liver to decrease the creation of glucose by glycogenolysis, further normalizing postprandial spikes in blood glucose levels. Because this effect is glucose dependent, reductions in blood glucose after the ingested nutrients are cleared will restore glucagon release and hepatic glucose production, thereby reducing the risk of hypoglycemia. The incretin system also regulates gastric emptying, intestinal motility, and satiety. For example, preclinical and clinical data suggest that high plasma concentrations of GLP-1 inhibit the contraction of intestinal circular muscle, which plays a dominant role in churning and moving nutrients through the small intestine. GLP-1 also binds to GLP-1 receptors on vagal primary afferent fibers in the gut. The activated neurons transmit signals from the gut to the brain, which relays the information back to various muscles of the stomach, reducing the speed at which nutrients pass out of the stomach and into the small intestine. This process helps spread out the intestinal absorption of carbohydrates, fats, and proteins over time, further blunting the size of postprandial spikes in blood glucose levels. GLP-1 induced activity in the vagal afferents also contributes to the feeling of fullness and reduced appetite following a meal. Although most of the incretin hormones produced in the gut are rapidly degraded by the enzyme dipeptidyl peptidase 4 or DPP4, some GLP-1 may reach its receptors in the area of the central nervous system involved in satiety. Moreover, some neurons in the brain express GLP-1, which may contribute to local pathways in the central nervous system that help induce satiety and indirectly decrease food intake. Clinically, GLP-1 analogs that are used to treat patients with type 2 diabetes have been designed to resist DPP4 cleavage. Therefore, when supraphysiologic doses are given therapeutically, these analogs likely stimulate both central and peripheral GLP-1 signaling pathways involved in satiety. Indeed, all the GLP-1 receptor agonists that have been shown to produce significant weight loss in clinical trials. A decrease in the incretin effect appears to be a specific and early manifestation of the development of type 2 diabetes. The underlying mechanisms are likely multifactorial. Deficient incretin-induced islet cell responses have been observed very early in the disease course as glucose metabolism becomes impaired before other signs of beta cell dysfunction are apparent. Some evidence also suggests that GLP-1 secretion after a meal is impaired in type 2 diabetes, whereas glucose-induced insulin responses to high levels of exogenous GLP-1 remain. Research into the details of these effects is ongoing. Conversely, type 2 diabetes is not associated with a major deficit in GIP secretion, but the hormone has little to no effect on pancreatic insulin secretion even at supraphysiological levels, which suggests that these patients are resistant to GIP. It is not surprising, therefore, that clinical research has focused on alternative agonists of GLP-1 receptors or products that inhibit the activity of DPP-4.